Hello everyone. In this lecture we are going to discuss about reducing disaccharides, especially the disaccharides which have C1 C6 glycosidic linkages. So in the previous lecture we have seen about the disaccharides which are having C1 to C4 glycosidic linkage. So in this video we are going to discuss about the disaccharides having C1 C6 glycosidic linkages, which again comes under reducing disaccharides. So the first one is Gentiobiose. So the Gentiobiose is a reducing disaccharide again composed of D-glucose molecules linked by a beta-1,6 glycosidic bond. So basically this Gentiobiose is composed of two molecules of D-glucose which are linked by beta-1,6 glycosidic bond, in which the bond forms between the carbon atom 1 and carbon atom 6. So this is the structure of Gentiobiose. These are the two molecules of D-glucose. So in the two molecules, between the two molecules, there is a formation of glycosidic linkage like this. So the glycosidic linkage is between C1 carbon atom and the 6th carbon atom of another D-glucose. So this forms a glycosidic linkage like this. So this is known as C1C6 glycosidic linkage. I hope you understood the structure. So it is naturally occurs in certain plants and is a component of important bioactive molecules such as crocin. So the compound responsible for the saffron's distinctive color. So there are some bioactive molecules like crocin is example for one such bioactive molecule responsible for the saffron's distinctive color. So it gives coloration, it enables the saffron to produce certain color. So due to its glycosidic linkage, Gentiobiose exhibits distinct physicochemical and biological properties that have drawn interest in biochemistry and biotechnology. Let's see the chemical structure and properties of Gentiobiose. The molecular formula of Gentiobiose is C12H22 and O11 with molecular weight of 342.30 gram per mole. So it typically appears as a white to off-white crystalline powder. Again, its beta-1,6 linkage between glucose units differentiate it from other glucose disaccharides like cellobiose. So here the cellobiose contains beta-1,4 linkages between glucose units, where in case of Gentiobiose it consists of beta-1,6 linkages between glucose units. Here comes differentiation point. So Gentiobiose is soluble in water and hot methanol and exhibits characteristic optical activity. So as it is reducing sugar and also it has certain physicochemical properties, right? So one such property is the exhibition of optical activity, and its melting point is approximately between 195 to 197 degrees Celsius. So I have mentioned about bioactive compounds, right? So there are some other kind of bioactive compounds are present. So basically these bioactive compounds are natural chemical substances found in small amounts in plants, like they may be present in fruits, vegetables and oils, promote good health. So one such example will be the crocin, which actually present in saffron. Let's see the biosynthesis and natural occurrence of Gentiobiose. So Gentiobiose is derived from the partial hydrolysis of gentianose or other glycans. So it can be naturally occurred, or it can be derived from the partial hydrolysis, half treatment, or half breakdown of gentianose or other glycans containing beta 1,6 linkages. So it can be produced enzymatically or chemically from natural plant sources, as it is present in plants as well. So this disaccharide is incorporated into larger glycosides in plants, contributing to their biological activities and colors as seen in saffron. Let's see the biological functions and applications. So Gentiobiose serve as a model molecule for studying carbohydrate protein interactions, particularly the binding of lectins to beta glucosyl units. It is utilized in biochemical assays related to glycosylation and in the development of sugar recognition therapeutics. Its unique structure has made it valuable for understanding the role of beta 1,6 linked glucose in biological systems. Additionally, Gentiobiose derivatives are explored for potential pharmaceutical and biotechnological uses. So here you can see the structure of Gentiobiose having beta 1,6 glycosidic linkage. So the second type of reducing disaccharide which having beta C1C6 glycosidic linkage is Melibiose. So here Melibiose is again a reducing disaccharide formed by alpha 1,6 linkage between galactose and glucose. So it can be formed by invertase mediated hydrolysis of raffinose, which produces Melibiose and fructose. So it can be produced by the hydrolysis of raffinose by the enzyme invertase, which gives Melibiose and fructose as intermediates or byproducts. Melibiose can be broken down into its component saccharide glucose and galactose by the enzyme alpha galactosidases such as MEL1 from Saccharomyces pastorianus, which means as Melibiose can be formed by the alpha 1,6 linkage between galactose and glucose. So if you can see the structure, so this is basically the structure of Melibiose, right? So the glycosidic bond present between the glucose and galactose. So the glycosidic bond formation between C1 carbon of glucose and C6 of galactose gives Melibiose, right? So if you again want to produce this glucose and galactose, when you break this particular bond upon hydrolysis again will give glucose and galactose as individual units. So this is how you can produce glucose and galactose, and you can produce Melibiose from glucose and galactose. So here, as I have mentioned, component saccharides glucose and galactose by the enzyme alpha galactosidase. So this broken down of Melibiose into glucose and galactose is possible by the action of enzyme known as alpha galactosidase, which break down this Melibiose into glucose and galactose. So this alpha galactosidase enzyme is coded by the gene MEL1, which is present in Saccharomyces pastorianus species. So it is widely studied in microbiology, particularly in differentiating yeast species, and plays a role in plant metabolism and advanced glycation end products. So this specific Melibiose helps is helpful in studying microbiological studies, particularly in differentiating the yeast species. So it plays an important role in plant metabolism as well as study of advanced glycation end products. So here, what are advanced glycation end products here? So these are harmful compounds formed when proteins or lipids or nucleic acids interacts with sugar molecules in non-enzymatic mechanism. So here basically the proteins which actually formed in the body will be linked to carbohydrates, will be linked to glucose to form glycoproteins, right? Linked to form glycoproteins. So this addition of sugar units to proteins is completely an enzyme dependent mechanism, which means the enzyme known as glycosyl transferases are present in this glycosylation process. But here, in case of advanced glycation end products, the glycosylation occurs, but it actually happened as an enzyme independent mechanism. So no enzyme is actually involved in order to produce the glycosylation products, which will leads to dangerous conditions. So here the harmful compounds formed when proteins, lipids or nucleic acids binds with sugar molecules in a non-enzymatic reactions. So it will leads to non-enzymatic glycosylation, which means no enzyme will be involved in the process of glycosylation. It will leads to non-specific reactions of glucose with proteins. It will leads to conditions like diabetes. You can see the excess amount of sugar in the blood, which will leads to diabetes. Example, glycated hemoglobin in diabetes. So this is one such example for the advanced glycation end products. So it is completely an enzyme independent process. So here the Melibiose has molecular formula C12H22O11 with a molecular mass of 342.3 gram per mole. It appears as a white to white of crystalline powder, often as a dihydride or monohydrate. Melibiose is soluble in water, moderately sweet, exhibits mutarotation, again intercombination between alpha and beta forms. So biological occurrence, as I have mentioned, it can be produced by the hydrolysis of raffinose. So Melibiose is formed during the enzymatic degradation of raffinose, a trisaccharide found in legumes, beets and molasses by invertase or beta fructosidase. So which means here raffinose is a trisaccharide, which means it consists of 3 units of sugar in its composition, where Melibiose is a disaccharide. So the production of Melibiose is possible from the raffinose by the action of enzymes known as invertase or beta fructosidase. So upon the action of this invertase, the raffinose will give rise to Melibiose, which is a disaccharide. So this Melibiose is found in some plants, seeds, leaves, stems and roots. So its application in microbial metabolism is many microorganisms can use it as a carbon source, but others cannot, which makes it useful diagnostic tool. So here the important application of Melibiose is industrially is differentiation. So in case of yeast, there are two kinds of species which we need to distinguish. If you want to distinguish between the two species of yeast, which are Saccharomyces pastorianus, which is also known as a lager yeast, and also Saccharomyces cerevisiae, which is an LE yeast. So if you want to differentiate between the two species by using Melibiose, here the lager yeast, which is Saccharomyces pastorianus, possess the enzyme alpha galactosidase or alpha galactosidase encoded by MEL1 gene would be able to break down this Melibiose, while Ellie strains, which is Saccharomyces cerevisiae, won't be able to do the breakdown of Melibiose. So this is how you differentiate between the species of yeast, which are Saccharomyces pastorianus and Saccharomyces cerevisiae. So as I have mentioned earlier, the Melibiose can be broken down by the action of enzymes like alpha galactosidase. So this alpha galactosidase or alpha galactosidase is coded by the gene MEL1, is actually present in case of lager yeast, which is Saccharomyces pastorianus, while it is absent in case of Saccharomyces cerevisiae. So for differentiation purpose, or it act as a diagnostic tool, the Melibiose is used in industries. So this is one such industrial application. So it is also act as a fermentation marker. Melibiose can be act as a fermentation marker. Its presence in fermented beer indicates it was produced via raffinose hydrolysis. Its absence suggests it was fermented by a non-Melibiose fermenting yeast. And also in transport mechanism, in bacteria like E. coli, Melibiose is imported to the cell wall into the cell via the Melibiose permease. This is a symporter that uses an electrochemical ion gradient to move the sugar. So in case of bacteria like E. coli, Melibiose can be imported into the cell by the action of enzyme known as Melibiose permease. So this Melibiose permease allows the Melibiose disaccharide into the bacterial cell wall. So basically this Melibiose permease is a symporter which actually transport the substances in the same direction by using an electrochemical ion gradient to move the sugar. So this is the structure of Melibiose again, glucose and galactose. So it has C1C6 glycosidic linkage. So this is the carbon atom 1 here, and this is the carbon atom 6, which will form a Melibiose. So these are the examples for reducing disaccharides which are having the C1C6 glycosidic linkages. I hope you understand this lecture. Thank you.